Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I'd like to invite you to mark your calendars for the very first Dr. GPCR Virtual Cafe held on March 26th, starting at 11 a.m. EST. Our first live virtual cafe guest is Dr. Brian Airy from Bristol Myers Squibb. Space is limited, so don't forget to get your tickets today. Visit drgpcr.com/virtual-cafe for more information. To present your original work or your company's technology, or to sponsor a Dr. GPCR Virtual Cafe edition, please fill out the form on the cafe's main webpage. The goal with the Virtual Cafe talks is to bring you GPCR-focused live presentations every month. You'll be able to join us live and interact with the speaker and attendees. We hope you'll join us. To stay on top of all your GPCR news, subscribe today to our monthly Dr. GPCR newsletter at drgpcr.com/newsletter. Last but not least, find us on YouTube. You can subscribe to our YouTube channel to watch the latest Dr. GPCR podcast episodes and much more.
Hello, everyone. This is Yamina from Dr. GPCR. I am delighted today to talk to Eleonora Comeo. She is our very first PhD student guest. She's from the University of Nottingham and Monash University. And we're very excited to have her here. Hi, Eleonora.
Hi, Amina. Thank you so much for having me here today.
I'm super excited to talk to you today. Thank you for coming on. You're the first PhD student, so I think this is an open invitation to all the GPCR trainees. Reach out, come on the podcast. I'd love to chat with you. So, Eleonora, tell us a little bit about yourself.
Right. So I'm Italian, and I moved to the UK back in 2017 to pursue my PhD. Well, actually, it wasn't mainly for that. The reason might be a bit more cheesy and romantic, but my partner had to move to the UK for work, and I graduated. I just finished my university in Bologna. I pursued my master in chemistry and pharmaceutical technology. So I was awarded Erasmus Plus for traineeship to work to do like a small period of research abroad, and I was really interested in the research they were doing in the lab I'm currently working on, such as Barry's and Steve labs. So they work on GPCR related. They do GPCR-oriented research, and in particular, I was really interested by the possibility of using fluorescent ligands as a means to understand GPCR pharmacology. So then I didn't think to do a PhD at all, and eventually I completely fall in love with that research, and I felt 3 months wasn't enough for me. So I asked whether they had a PhD opportunity, and then I did the interview, and here I am. So it was really nice.
That is great. And how did you find the lab? So you were doing a master's at the University of Bologna. Were you working on GPCRs? Have you been exposed to GPCRs by then?
No, at all. I started working on GPCR in Barry's lab and Steve's labs. But at the university, I started only the basic pharmacology because I'm also like a pharmacist by training. But of course, you study the basics. You don't really go in a deep understanding of the research that is done around that protein.
Yeah.
But I really love what the information that you gain by using these tools as well, and I felt I wanted to pursue this a bit more, so that's why. And in particular using also the medicinal chemistry part, because I'm a medicinal chemist, so the fact that I'm making those molecules and I can see them in my hand, that's really like a really fun fact of working in this specific research field as well.
That is great. And just to dig deep a little bit more on the practicality of it. So you decided to go, well, you were following your partner to the UK. And did you go online, look at different labs that were there? how did you go from the moment that you decided to follow your partner to the UK to the moment that you joined the lab and started working in there?
After a couple of months, because I was still in Italy, so I was looking for various lab around the area where I was working, and again I found various lab in Nottingham. It was really nice. Well, it was like 1 hour distance, driving distance from in office. So I found, well, it still seems like we can work this out, and then yeah, I found Barry's lab, and he was really happy to having me. I was also surprised because I didn't have like a huge background in this field, since I was like a chemist and a pharmacist by training, but I've never really done research in this specific field. But yeah, it gave me the opportunity to start fresh, so it was really nice.
It's great. The reason I was trying to get to understand how you went from looking for the lab to finding the lab and ultimately joining it is because I think when you're a student, a master's student, or early career scientist, sometimes people may feel a little bit shy
Yeah,
to reach out to PIs, and or they tell themselves, like you did. You had just mentioned that you didn't have experience in the field, but you did still go for the interview and you went for it. And I think that is something that is a great message to put out there to all the trainees. Just because you don't have the background doesn't mean that you shouldn't apply, you shouldn't get to know the scientist.
yeah, most definitely. Especially nowadays, I think that you can just Google everything, right? So you can get access to every research group webpage, but not just in academia, even in industry. Well, maybe in industry sometimes it's more tricky, unless if you look for small companies. But I mean, it doesn't really hurt to try. And I mean, the worst case scenario, you get a no as answer, and then you try the next one. And sometimes I guess it happens in life. It might be that it's just part of your path. That wasn't the right time for you to go there, but you needed to go somewhere else. And so yeah, I've always tried to follow also, well, my brain, but my heart in this sense. Just I mean, looking at what's the worst option. So just try.
Exactly. And you have nothing to lose when you just send out an email, and if you get a response, something magical may come out of it, and you may fall in love with GPCRs.
Yeah,
And having a medicinal chemistry background
yeah,
allowed you to take that knowledge and apply it to a brand new field,
yeah,
which
yeah.
I think it's really valuable. The other question I wanted to ask you is, so you went to the University of Nottingham. Where does the connection to Monash University come from?
So, because the program that was available at the time was a joint program between the University of Nottingham and Monash University, so which is really great because you have the opportunity to spend 1 year abroad. And I must admit that at that time it was a bit scary because Australia, of course, is on the other side of the world, so only the thought to spend 1 year there alone was a bit scary, but at the same time really exciting because I went there on my third year, which I think is the best time to go there, because or to go in a different lab, because you have the opportunity to start again, even though you do research in a similar field, because of course it needs to be something relevant to your thesis. But it allows you to challenge yourself again, because you go in a different lab, so you meet up with new people and you feel you need to prove again your abilities, and also you just meet new people and potentially new partners in the future. That's one of the reasons I like doing research, is that the fact that you meet up with different people coming from different countries and have different backgrounds. So I feel really rich every time I talk to them and kind of learn something new.
That's a great experience, and especially going to a lab that's at the other end of the world. I mean, I'm in Boston currently, so there are 12 hours or 13 hours ahead of us, which is just fantastic when you think about it. And what I loved, that you had mentioned that you were happy to start fresh in a new lab and meet new people. I think as a PhD student or as a scientist, when you start working in the lab, you develop skills beyond the science. So you learn how to think, you learn how to ask questions, but you also learn how to collaborate and how to work within different teams. And I think that is valuable as being, hardcore scientist, because that what takes you to the next level, independently of where you want to go once you finish your training.
Yeah, yeah, exactly. I think is part of our training. I guess, well, in the end, a PhD is different because it's, even though you pursue a scientific pathway, but is a personal pathway. So everyone experiences this in a different way. Um, but I think, yeah, it's just really nice to change sometimes, and you need a bit of a like a spark, and to put yourself in a different environment, because that's what happens in life. Sometimes I guess you find yourself, you need to change maybe a job because there are no current opportunities. So you just need to be plastic.
Yes.
So yeah.
It makes you way more flexible.
Yeah.
Let's get back to the science. Can you tell us a little bit more about what does your group in Nottingham work on?
Yeah, so in Nottingham, as well as in Monash, my group works mainly in GPCR oriented research. It doesn't involve only one specific receptor, but many of them. In my field, I've been interested in looking at the pharmacology of adenosine receptors, with particular focus of the A2A and the A1 subtypes, because adenosine receptors are a family of four member of receptors. However, my group does research in other sort of GPCR, such as histamine receptor, adrenergic, beta adrenergic receptors, and chemokine receptors. So it's pretty broad. And even in Monash, it's not just the GPCR, but they try to develop drugs even as anti-malarial agents or anti-cancer. So it's pretty vast, and you get exposed to different kind of research as well, which is quite interesting.
Great. And how big are the labs?
Um, the lab are quite big, and I mean, of course, are different. Like the chemistry lab. One of the things I like about my project is that, of course, I'm not only doing the chemistry, but also, I'm also doing my own pharmacology. So I'm experiencing both part of like development of these molecules, and the labs in chemistry are quite big. Everyone has his own fume hood. Well, not now, unfortunately, because of COVID. So there are a bit more tighter regulations. So
Mhm.
there is a smaller capacity allowed in the lab. So we work in shift, and also not all the fume hoods are occupied. But in theory, everyone has his own allocated fume hood. So you do your chemistry there. Whether in the pharmacology lab, there are like open benches, because you don't, unless you work with toxic chemicals, so there
Yeah.
are fumes there as well. But and the pharmacology lab are quite big as well.
So there's a chemistry branch of the lab and a pharmacology branch of the lab in
Yeah.
both Nottingham and in Monash.
Yes, in particular in Nottingham, they are in separated buildings. So in Nottingham, we have the Biodiscovery Institute. There are like three main buildings where we don't only do medicinal chemistry research, but there are also pharmacologists. There is molecular biology research as well, different kind of research. And then the pharmacology lab is allocated near Queen's Medical Hospital.
Mhm.
And so I just have to cross a footpath bridge. Sometimes I was running back and forth from the two labs doing chemistry maybe in the morning, then I had like 2 hours time and was going back doing tissue culture. Uh, and it's quite interesting. I need to go to the gym because it just do so much activity in the labs. Whereas in Melbourne, there is like a big institute, so it's the Monash Institute of Pharmaceutical Sciences, where there are different floors as well, and with different departments. So I was just simply going upstairs, for example, when I need to do my own pharmacology.
That is great. I can just imagine you, putting on your chemist hat, making the molecules. It's like you're making the ingredients to actually test them later in cells, which is just phenomenal. So you mentioned that you work on adenosine receptors, and 2 out of the family of 4. Can
Yeah.
you say that these would be your favorite GPCRs?
Yeah, absolutely. I know I'm completely biased because it's my PhD program, but I really, as soon as I started to understand, to the best again, what we can understand still about this receptor, I really like it because um, those receptors are virtually involved in all the physiological and pathological process happening in the human body, and there is potential for targeting this receptor for many diseases as well, and they are super interesting. Unfortunately, though, one of the main disadvantage of this receptor is the fact that they are so involved in many pathological and physiological conditions that makes this receptor really difficult to target. And in fact, there are so few drugs in the market available using the clinic that target this receptor protein. Adenosine itself and one like a drug agent which is used as a diagnostic agent is called regadenoson, and then which is an agonist for the A2A receptors. And recently, actually, I was quite happy because last year, after many years, for example, the A2A receptors is involved. It has been studied a lot because it's involved in Parkinson, and due to its colocalization with the dopaminergic receptors, it has been targeted as a way to treat the symptoms of Parkinson, because it seems that if you block the adenosine receptor, you enhance the motor activity in patients with off episode of Parkinson's disease. And finally, last year, this drug has been approved after many years of clinical trials, so by the FDA. So that was quite a big news.
Of course, any I feel like any new drug targeting a GPCR and that gets approved by the FDA is a big news, and it's great. I think we're all somewhat working towards that in different ways. Some in the lab, some in biotech pharma, and me doing these interviews with you so that we can share this information. So you had mentioned, can you tell us a little bit more about the pharmacology of the adenosine receptors? And you had mentioned that they're involved, expressed almost everywhere in the body, and that they're involved in many diseases. Can you just give us a little snapshot about adenosine receptors?
I will try my best. So the adenosine receptors, as I said, are 4 members. So A1, A2A, A2B, and A3. A1 and A2B are more like characterized and more well studied, where the A2B and the A3 are still under deep investigation. What is interesting about these molecules that, they're G protein couple receptors, so there are a couple with different G proteins. So mainly, for example, the A2B and the A2A are mainly GS couple receptors, so which means that they activate adenine cyclase and enhance the cyclin P accumulation, where the A1 and A3 are mainly GI couple. Although what is becoming pretty obvious is that they're not just the fact in also different tissues, those receptors can be coupled with different G proteins. So a major problem in targeting this receptor is not just the selectivity in specific receptors over the other, but is also the G protein selectivity and the tissue selectivity that is a major problem. And this also, I guess, represent one of the challenges for the development of therapeutic agents that target these receptors. And what is interesting also about, and I like it about these receptors, that for the A1 and the A2A, there are really many crystal structure. Well, particularly for the A2A, there are over 30 crystal structure that has been solved. I think I might have counted them one, might be around 42, 43 if you look at the protein data bank, which definitely allows deep understanding of these receptor. In particular for myself as a medicinal chemist, the design allows me to try to rationalize much more the design of drugs and fluorescent ligand that can specifically target these receptors.
Wow. And so you're just mentioning that there is four adenosine receptor subtypes.
Yeah.
And what's the difference between A2A and A2B? I know it's a naive question, but because sometimes the receptors get named as they get discovered, but these two, do they have anything in common?
Well, yeah, they both activate mainly GS coupled. However, it seems that the A2A receptors is known to be like a higher affinity receptor for adenosine, which is, I apologize, I forgot to mention, of course, our adenosine, because our name after the endogenous modulator, which is adenosine.
Yep.
And seems that these are being activated by high concentration of you need not much concentration of adenosine to activate these receptor, whereas for the A to B, they are kind of known to be low affinity receptors because they need to be activated by a high concentration of adenosine. But I think also this distinguishing those two based on low affinity, or affinity is still a bit maybe resistant naive. So there is a bit debate on these as well. So, but and the A to B is particularly involved in cancer as well, and the A2A, the A to B in particular. So there are quite a numbers of clinical trials involving antagonists that try to block these receptors because those are expressed in immune cells, and in particular in the tumor microenvironment, where the amount of adenosine, the concentration of adenosine is quite high, and then this concentration is enough to activate these receptors that are expressed in immune cells, and it has been observed that the activation of this receptor kind of silence our immune response, and the tumor is kind of able to escape the immune response and to start growing and diffuse. So there are a couple of clinical trials now going on with selective A2A ligands, but as well most recently a dual agent that acts at the A2B and A2A as well, acting at both receptor because they are implicated in these pathology. I'm kind of hoping now that those two receptors also would be more validated in immuno-oncology, given the great promise that they have in this field. So just, I guess, we need to keep our fingers crossed, see they don't fail again.
How cool is this? So you have two receptors of the same family, and they have two different affinities to the same ligand, which usually when I think about bias, I think about the traditional bias when you have one receptor and then you have a G protein versus beta arrestin type of bias, but this is just two receptors of the same family with different affinities, which is, I think, it's a beautiful way for the body or the cell to control a response to adenosine.
Yes. Another thing I think it's worth pointed out is also that, of course, the affinity also depends in which experimental setting you measure the affinity, I guess.
Yeah.
Is whether you measure in a cyclic-MP kind of assay or whether you measure another response, it might be that the affinity, of course, is different because it has been actually observed that if you measure the affinity of adenosine at these receptor subtypes in a different assay, eventually similar to all these receptors. Very few measure a cyclin MP response, reiterate the difficulty in targeting this receptor and addressing important questions.
Can these receptors be expressed in the same cell type in the body usually?
Yeah, that's another. That's another problem. For example, X cells, they endogenously express A2A and A2B, actually quite high level A2B receptors and A2A. That's why I guess when you, I remember one of your first podcasts where Terry discussed about X cells and looking at the cellular environment. It's really important, and especially even for myself when I go and test my fluorescent ligand, and of course I don't know yet whether they're selective. That is what I want to achieve. It's really important to understand and to know also in your cellular context what's the expression of the other receptor, in particular the receptor you're interested to study, because otherwise you can get confounding outputs. So, yeah.
That's the importance of having negative controls.
Exactly.
I once went down a path where, so I was working on actually two different receptors important in B cell development. So one chemokine receptor, CXCR5, and the other one was a sphingosine receptor, so S1P1R. And I was trying to put together these cyclic AMP assays. And what I noticed is that, so there was no background for CXCR5 in hex cells, but there was a huge spike with just cells, non-transfected cells with the receptor for S1P ligand. And it turned out, so I went down this whole rabbit hole where I realized that out of the 5 S1P receptors that exist in nature, there were 3 expressed in hex cells.
Oh.
So it made it very difficult or almost impossible to use these cells. And the rabbit hole started out when I said, well, let's just make CRISPR. Let's just CRISPR out all the receptors we don't need. And unfortunately, I didn't get a chance to test them. But yeah, I think that's an issue. And as you mentioned, Terry also, and I think Graciela also had mentioned in the podcast about the hex cells, and they're easy to culture, they're easy to use. But at the end of the day, if you want to move and understand the receptor function in their native tissue, at some point you have to move out of hex cells.
Yeah, definitely. So this is, I guess, would be one of the next steps. What in our lab they do as well is like because we develop also these tools. So of course we need to study these tools in a really deep way, but then we want to see whether they work in a endogenously expressing system, because that's what you want to understand. I guess when you start maybe doing a PhD at the beginning, you need to learn the techniques. So of course you go and you are a bit more focused on standard methodologies. But in the end, you need to progress and have a deeper understanding of the function of these protein in more relevant setting. And
So tell us more about the tools, about the fluorescent ligands that you synthesize, which I find fascinating. I am not very good at chemistry. I always thought it was magic. It feels like magic to me. So tell us more.
It feels a bit, because one of the really interesting thing, actually I remember from the my interview, that Steve asked me, why it's really interesting using these tools. And so I remember I was still thinking, what kind of weird question or answer I have to do, like I have to give like really technical one. And then it's just because you can see them. So they're really useful because you can see them and you can use them to visualize the process. They're interesting too. The really nice thing, well, of course, the chemistry in every field is not as always easy. It's quite tricky, and sometimes because you need to modify to make those compounds. Of course, you need to identify like depending what process you want to visualize, you need to modify agonists or antagonists for the protein you want to study. And usually we try to look at what's the best compound available based on the literature and most selective potentially. And then you look at how you can modify this drug. And for the A1, A2, A receptors I mentioned before, there are crystal structures. So there is structural information that they guide us to the synthesis of this drug, these molecules. And so then we, after that, we analyze it, and then we start synthesizing, and we add a linker potentially, and our fluorescent tag of different colors. So there are fluorophores that cover the whole spectrum of light. And so you select that one that for you it can be the most optimal for depending on the application. Usually for us, we tend to prefer like far red emitting fluorophore because they emit in a region of the spectrum where there is like less background autofluorescence coming from the living cells. So there is the information and the signal to noise ratio is lower, sorry. And so one of the beauty is that you do your reaction in this really tiny vial because, of course, you don't need much of these compounds, especially when you start screening them. And I tend to do my reaction in small CMS vials.
Oh.
And then you get them, because, of course, they're colorful. So they're blue if you use like red-shifted fluorophore. And so you go around with this blue solution, and then you purify it, of course, and then you start making your aliquots, and then you start testing them, hoping for the best that they are active and do the job. But one of the things I like most, of course, is doing the chemistry, because I love doing chemistry. But in the end is after that you go through the process of testing them, because it's quite a long process. First of all, assessing the pharmacology. This is really important when you develop tools, I guess, in general. You need to assess whether they're selective, whether they are functional, agonists or antagonists for the protein you want to study. And then eventually you use them, for example, in confocal microscopy study. And you go to the microscope and you see they bind, and you see the nice labeling of the cell at the cell membrane, because, of course, adenosine receptor at GPCR so are located at the surface. And it's really rewarding in one sense, because you can actually see in practice that these tools are really useful. So.
And can you, since these ligands are fluorescent, can you follow the receptor once within the cell once
Yes.
they bind? Wow.
Definitely. Actually, it is one of the powers of this technique. The A1 is a bit more complicated because they've shown to be really slow in internalizing, but you can definitely use fluorescent ligands to do that. As well as NAP technology, of course, you can just use NAP tag A1 receptor or A2A receptor, and then you label them with a fluorescent tag, and then you can study them function. And in particular with like a super resolution technique, you can have a really clear information of the receptor, how this receptor respond in the single cellular level, and also in the different compartment, because you can have that great resolution in combination of the technique, fluorescent technique, and the fluorescent ligand as well.
That's so great. So basically, if you had to, from a high level, think about your contribution to the field, it would be developing these tools to better understand the pharmacology.
Yeah, that's kind of the overall point, because one of the key things of, well, all GPCR really, but in particular the adenosine receptor, that seems to be, despite the great advancement done, so there are really like extensive number of ligands developed for these receptors. They show to be really good in terms of affinity, selectivity. Then they go all through the process of clinical trials, and then they fail, either because they don't show efficacy for the drug, for the receptor of interest, or because they start developing side effects, which is particularly problematic, especially for the A1 receptor, in terms that the same receptor is involved. So the activation of the same receptor can also cause the onset of side effect, because all their pathway signal activated. And so we can use different approaches to target the receptors. These for example, it can involve like biosignaling or development of allosteric modulators. But of course, these highlight the importance of having a tool that can allow us to investigate this receptor in the local environment. So in an environment that is not modified potentially.
Yeah. So you'd mentioned that in HEC cells, 2 or 3 of these receptors, actually A2A and
A2A,
A2,
particularly A2B,
are expressed. So when you test your fluorescent ligands to adenosine receptors, what how do you circumvent the fact that, what do you, first of all, do you use HEC cells, and second, how do you circumvent this endogenous receptor expression?
Yeah. So well, what I've done even in the past, the reason why we use cells, because a human cells, so you want to have like a cellular environment that kind of represent a human kind of environment. And in particular in our group, we use nanonuc tag technology. So we use nanobreath, which involves the use of receptor, which is tag at the end surface of the receptors, at the end terminus of the receptors with the nanolook. And usually I express this receptor in X cells. And then we use we do standard binding experiments such as saturation binding, competition binding experiment, and then as a control, of course, we use like an unlabeled antagonist that blocks the response of these fluorescent ligands, for example. And you can actually see that if it's the receptor, because, for example, if you use a selective A2B antagonist or A2A antagonist that blocks the response, it means that your ligand is eating those receptors as well. So you tend to use this control, and in particular, even for imaging studies, of course, again, we use system. In my imaging study, I use like SNAP-tag A1 receptor, for example, and I've also carried out studies in non-transfected X cells to see whether my fluorescent ligand were actually allowed to visualize any sort of labeling. And we have actually showed that it doesn't happen, but of course it can happen when you have a non-selective fluorescent ligand. So it's good to have it's definitely important to have a control, especially if you want to develop a tool that needs to be properly used to understand the pharmacology. So you don't want to have facts that can confound your reading. So yeah.
Again, the importance of having the correct controls when
Exactly.
when you do these experiments. Are these receptors known to form dimers by any chance?
Yes, they form dimers, and they've been quite investigated, and has been shown by crack technology, really a group in Spain a lot, and are researching these forming, showing actually one of the first that showed the fact that A2A was forming this dimer with the dopaminergic D2 receptor, and so the importance and the involvement of this complex, for example, in Parkinson.
Mm-hmm.
However, they can form, heterodimers, so with different proteins and receptors, as well as homodimers. I think although there is still a need to understand potentially the biological implication of this all these different heterodimers in the function of these receptors. Another thing that my group in Monash, for example, they're investigating is the involvement of the dimer formation between the A1 and A2A receptors in cardioprotection, for example, because the activation of adenosine receptors mediates cardioprotection, and is being investigated for development of treatments for ischemic heart disease. So it's still under investigation.
Wow. The reason I was asking about the dimers is since you do have, and this is nitpicking, this is me just like really picking on the HEC cells here. So you had mentioned that you do have endogenous adenosine receptors expressed in the HEC cells, and then you transfect your nanolook tagged receptor, is there any chance, and I'm sure you thought about this, is there any chance that your transfected receptor forms a dimer with the endogenous receptors, and would that alter the response that you get?
This is a tricky question, but I think they could be a chance. Would be actually quite interesting to investigate these, and to have this answer. I think there could be a potential. Maybe not with A2A, but yeah, it would be interesting to investigate.
I always wondered these things. So yes, we agree that using HEK cells, it's easy. They work well. They grow like grass. It's easy to work with them, and you do get your answers pretty quickly when you work in HEK cells.
Yeah.
But I always wondered if the receptors that you transfect in the HEK cells interact in any way with any endogenous GPCRs that are in the HEK cell, and would that alter the response? I don't know. I mean, it still would reflect the what happens in a native cell in the body, because cells in the body also can co-express HEK cells. I mean, GPCRs. But still, I wonder if that would, how would that change? We don't necessarily have the answer to that question, but I thought I'd ask.
No, I think anyway, every time we tend to use not just HEK cells, any kind of cellular environment, and we exogenously express the receptor, it's going to be a kind of alteration on the own pharmacology, because simply just the forming like maybe homodimers between the same receptor might alter the pharmacology or your readout anyway. And this has been actually investigating even recently in our lab, where they have developed this cell line using CRISPR-Cas technique to endogenously express the nanolook, for example, A2B or chemokine, on the chemokine receptors, where you can actually get the response under endogenous promotion, and then you can actually study this with nanobread. And it's quite interesting because you get, even though the expression, of course, is kept low, like endogenous level, you can get really like a really nice signal. And I think this actually opened up new opportunity for study these receptors at endogenous level in combination with fluorescent ligands. And you can have a better understanding how this receptor works.
Yeah, that's super. So are you finishing up your PhD?
Yes.
Great. When are you're writing your thesis right now?
Not well. I guess I'm finishing. I have 1 year left, but I'm still. I still want to stay in the lab as much as I can because I really like being in the lab. So I guess the plan will be to finish off these couple of projects I'm working on, and then start writing my thesis. I have to submit by the end of next year, September. So yeah, it would be. I'm a bit sad, to be honest. I mean, I know when you have fun, of course there is a beginning, there is an ending. But I really love this journey so far, and it's scary, but at the same time, it's the right time to start a new career. And I'm also looking forward for what like the future has to me. So
that's super. So you're quite experienced, and what are some of the take-home messages you'd like to give someone who's just starting their PhD or is thinking about doing a PhD?
Well, I guess one of the first thing is not give up, just be resilient. And I know it's difficult. I know it's personal. It's a personal path, but I'm sure if you like what you do, there will be a time in your path where you can get you can retrieve all your success, and also try to talk with your peers a lot. Like one of the thing I've discovered, of course, the PhD, you do your own project, but what I like the most is just going around and talking and trying, be curious what other people work on, because it might be even that they work on a different project, but they might have like encounter in a problem that you're facing right now, and so they might be able to talk to you, and also you might be able to just find new collaborators, and just being honest and enjoy your pub, because it's kind of your baby as well, right? A bit. Yeah. So yeah, a PhD is different, but I think is important. Especially another thing is collaboration, because sometimes you can feel a bit of lonely, because you're working on a project that in theory you're trying to solve like a big question, right? But there's always someone that can help you. So another thing, don't be worried or scared to ask for help, because it's fine to not know the answer. That's what you're trying to answer. And I found this a bit difficult when I started, because you finished as an undergrad and you kind of get to know that sometimes not knowing the answer is not good, because you need to get high marks at the exam, so you need to know the answer. So the transition from undergrad to a PhD, when you are actually trying to find the answer to your question, is a bit tricky. But I think as long as you admit everyone has his own limit, so yeah.
I think you make a great point about, communicating and asking for help, but also talking to people, because you never know who can tell you, oh, I've tried this and it doesn't work, or I've tried that and you should be, I don't know, doing something specific to make your experiment work. And it saves a lot of time, a lot of headache, and a lot of energy as well. I don't think you should spend too much time in your corner and bang your head against the wall. Just go out there, take some time off, think about the problem. I've had the best ideas in the shower or on a walk.
Exactly.
And I think that's very important. And I had the best collaborations come out of just plain discussions on problems that I was facing, or potential collaborators were facing. And it was just what we did. We just sat down for coffee and I was there to help, or the person in front of me was there to help me. And it ended up being a great collaboration and creating a great relationship as well with fellow scientists.
Yeah, definitely. It's important to have like this balance, and even in chemistry, sometimes like you think again you're working on only one scaffold, one molecule, and so you think no one has done that kind of research. But it's just asking. And in our lab, in our group, we kind of used to have this whiteboard, so we used to go there and brainstorm together. And because of course there are people coming from different background, have different experience, so they're definitely going to give you some answer that sometimes even like all the research engine are not able to give you, because maybe you're not type the right word. And the experience and just talking with these people is really really important, and I think is what may helped me a lot even in my PhD. Just not be afraid and talk with your PI and with your mentors and just your peers as well, and just make new friends anyway. So that's really good.
With the new COVID situation and going into quarantine, would you say that the ability to talk to people openly about your work and your difficulties is something that you really miss?
Yes. And another thing that I miss as well is just walking by in the lab or in the corridor, because we wear masks, and I used to smile to people. Whereas now you kind of tight your eyes, and so people might think, "Oh, what she's doing," and so that's fear, something I really miss. And again, some of the things, just simply doing our problem session together, so
Yeah.
being in a room all together, all those kind of things, we can't do that. As well, just having a meeting even with my supervisor, being able to see his like real face, not through a monitors.
Yeah.
And another thing, I guess, conference. I mean, despite us being really great, all the conferences, even you organize such a really great opportunity that sometimes we wouldn't have. I think we miss the actual networking opportunity sometimes, just going around and being able to go and talk maybe with the specific speaker, or maybe that one comes to your poster, and
Yeah.
it's a bit different. So this kind of opportunity now it won't come back. Well, for me and for whoever is at this stage, maybe, which something it's a bit unfair, but it's unfortunately.
Yeah. Yeah.
It's
I think what I would miss if I were, a PhD student or, a trainee, I think it's that awkward minute when you're at a conference and you sit down at the dinner table with people you don't know, and you introduce yourself, and then you realize, oh, this is that speaker from this university. But then it turns out that having that courage of sitting down at that table and getting to know personally these scientists is just very enriching. Let's flip it around. Is there something that you can think of that this whole COVID situation has brought on that you think is a good thing, that you took something positive out of it? Because I want to end on a positive note.
yeah. Well, as a positive note, as I said, I've been actually attending many conferences online, so that's still the thing. So I've been exposed to different kind of science as well, and maybe in other circumstances I wouldn't have even the time because I would have been in the lab. So this has been really nice, and maybe try to keep updated with my writing as well, in terms of practical positive aspect. And also it made me reconsider a lot the importance of having like personal life as well. Sometimes I guess, especially when you work really hard and you tend to just be focused on your work and forget a bit about maybe your family and your friends, whether under these circumstances you actually understand that not only science matters, but it's also, well, we are not just our work or our science, despite we love it so much, but our personal life is also really important. So that's another thing.
I'm glad you mentioned this. So how has your how was your work life balance before COVID, and how is it now?
Um, well, that's a tough question because I think it depends on the people. Personally, I'm really like stubborn in terms that when I'm focused to achieve something, I'm not really happy until I try to get it complete, especially when I like it. So it's not just that I do it because I have to, but it's something that really keeps me involved. And I've never really minded to go in the lab even the weekends or stay longer, because if something I can finish now, why I have to wait till tomorrow? But of course, that is different among like different people.
Yeah.
I think definitely that you need to be to have a bit of spark, but also the hard work in the hand in the long run will help you to achieve your goals as well. And especially given this time that now we don't have enough time in the lab, I actually miss it a lot. It's like but it's important to have a good balance because we are not machines, so we need something. And also, as you said before, the best ideas just comes out from not doing deep research in the literature, but just simply talking to people. Or even I do love running, and I think that I miss and going to run. There is this park run, which is a huge community in the world where they organize this group where people run like 5K and is everywhere in the world. So I miss doing that because, of course, we can't do that. So I usually go by myself. But when I run, I actually think about everything. But sometimes I got really nice idea as well. maybe about my chemistry wasn't working. So I started thinking, oh, what I can try to fix. So it's good to have something to escape. Yeah.
Yeah, I mean, I think you touched on a very important point there as well, where you cannot be always working.
Yeah.
And being 12, 14, 16 hours in the lab is not productive at the end of the day. Sometimes you have to do it. You have to finish a set of experiments. You're responding to a reviewer. That is fine. But I think having that separation between the lab and your personal life is very important, because it'll help you last as long as possible in the lab, but also will give you the opportunity to your brain to process what has happened. And it's funny that you were mentioning that you don't mind going into the lab, and you really like to finish things. And I think all of us as PhDs or as scientists share this dopamine rush.
Yes.
I mean, it's that moment when you've been working for months and it finally works, and you've been there for 10 hours and you get the final result, whether it's a number or it's a picture or it's a graph. It happened so many times that it was 5 o'clock or 6 o'clock on a Friday night, and I was like, no, I just need 30 more minutes because I need to analyze the data. And 2 hours later you see your curve and you're like, yes, I did it. And I think that's the beauty of it. And I think that's what makes scientists last so long and enjoy science. It's this need for discovery and the questions that we ask ourselves. But at the end of the day, when you think about it and you reduce it to that dopamine rush, is just priceless.
Yeah, it is. And I wanted to point it out even before. It's just like in chemistry, it's like when you see finally after months of struggling, you see the NMR of your structure without anything but your compounds. It's just absolute perfect. The rush, or even like you can feel the shivering in behind your spine as well. It's just really, you would like just go and run around screaming finally.
Yes. And usually it happens when you're alone in the lab and you have no one to share it with.
Exactly.
But it doesn't matter because you did it.
Yes.
That is so great. Thank you so much for your time, Eleonora. It was great chatting with you. I wish you the best for the next steps in your career. Take your time, finish up your PhD, enjoy it, and just enjoy this time. I think whatever comes next, you still have this beautiful experience, and you seem to have a really, a passion for chemistry, but also a passion for pharmacology. So great talking to you today.
Thank you. It was really nice being here. Thank you.
Thank you.
Thank you for listening to this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Don't forget to subscribe to the Dr. GPCR newsletter at drgpcr.com/newsletter. Until next time, stay safe.